Hi, I'm Dr. Ken Sharlin. I am the chief neurologist and owner of Sharlin Health and Neurology Functional Medicine. I want to welcome you to this video today. We're going to talk a little bit about my regenerate pillar and a little bit about stem cell medicine as well. I've been creating a series of videos that focuses on what we call the 5 pillars at Sharlin Health and Neurology. And this is our way of approaching problems that affect the nervous system, that affect you. What do I mean by that? We start with correctly identifying the problem. Then we want to investigate the root causes. Then we're going to help you to integrate all those root causes identified into a single cohesive plan that you're able to execute. We also have a restore pillar that focuses on the importance of hormones and brain health and ultimately helps you become the best version of yourself. And finally, we have the regenerate pillar that focuses on our proprietary stem cell approach using what are called medicinal signaling cells or mesenchymal stem cells. They're the same thing. I will elaborate on that a little bit more in a moment. I have another laptop open in front of me. I'm going to be using some notes as I go along. This is a fairly complicated topic, but I'll condense it down for you and hopefully make it easy to understand. I have a more detailed presentation I'll also be putting together and invite you to join me on that pre-recorded webinar in the near future. To start off with, it's important to understand that we are in the age of cellular therapy. Cellular therapy or biological therapies are used throughout neurology and rheumatology, as well as cancer treatments. Sometimes these approaches involve manipulation of the immune system to, say, attack cancer by focusing on your own immune cells and the behavior of your immune cells to go after the cancer rather than killing the cancer itself. Sometimes it focuses on treatment with a variety of antibodies. These antibodies are very similar to the antibodies your own immune cells produce, and they act like a very precise key that goes into a lock because they can be designed to target highly specific areas on a protein that then in turn affect how the cells behave. This might, for example, treat a disease like Alzheimer's disease, or in other cases, shut down migraines from where they're starting. Finally, we can use cells that are introduced in the body, and that's where stem cell treatment comes in. These stem cells are normally found in places like the bone marrow and fat tissue in the body. And when there is injury and inflammation, this is our first line of defense. These cells come out, they are anti-inflammatory, and they participate in tissue repair. So let's look at this in a little bit more detail. What is a stem cell? A stem cell is a type of cell that is a non-specific or non-specialized cell in terms of its function. They have the capacity for self-renewal, and this gives the rise to more stem cells. They're capable of differentiating into different lineages, different types of cells under appropriate conditions. And basically, there are 2 types of stem cells. There are embryonic and non-embryonic stem cells. And our focus is going to be to talk about the non-embryonic stem cells. But first, I need to give you a few definitions. You'll hear these terms throughout discussions of stem cell medicine, published in blogs and talked about in podcasts or summit recordings or journal articles, of course. So it's important to have somewhat of an idea of what these words actually mean. The first two words are autologous versus allogeneic. Autologous or auto means self, so the stem cells in autologous transplants come from the person who will actually get the transplant. In other words, the patient is the donor. Allo, or out from allogeneic, means other, so that the stem cells in allogeneic transplants come from a person or an animal other than the patient, either a matched or an unrelated donor. At Sharlin Health Regeneration Center, one of my sister companies, if you will, we only focus on autologous stem cell transplants. There's another term, institutional review board. This is an independent ethics committee, an ethical review board or research ethics board that is responsible for reviewing the methods proposed for a research study to ensure that they are ethical. We see IRB approved as an expression thrown around a lot on various websites and companies that promote stem cell treatment. So it's very important when you hear the term IRB approved that you understand that this does not mean FDA approved. This just means that an institutional review board has gone over the protocols and the ethics, if you will, of performing a study. So this is still research, this is still investigational, and given their stamp of approval to move forward with the research study. Again, very important to understand that IRB approved is not equivalent to FDA approved. Another important term is investigational new drug, or IND. Investigational new drug. This refers to a drug or a biological compound that's been approved for use in a clinical trial to investigate its safety and or efficacy. So when we use the term IND status, that means that it's been approved for use in specific clinical trials, okay? So if you have IND status for a study, for a compound that you're investigating, you can't use that in a completely unrelated study. You have to have separate approval for each study that you are doing. There are stem cell companies that have IND status for their product to treat COVID-related lung injury. That does not mean that their treatment is approved on an investigational basis to treat Parkinson's disease, for example. Now, there are different phases of clinical trials, and we'll run through those because, again, I'm most interested in you guys getting accurate information when you look at websites, when you hear about clinical trials, when you read a blog, when you listen to a podcast, when you listen to a summit speaker, because sometimes the language can be and I honestly believe that sometimes it's intentionally confusing, and that's the problem. So what are the phases of a clinical trial? Clinical trials ultimately are about getting products out to market to you so they get commercialized under the US healthcare system, right? So you, for example, see drug advertisements on television, and those are now available because they're FDA approved, but they've gone through all of these steps. So it starts out with a preclinical phase where the drug is being studied in animal models or in cell models in the laboratory, in petri dishes or fruit flies or mice and so forth. There is still ongoing animal research, although of course some of that has been limited in more recent years by animal rights advocates. There are phase 1 studies. Phase 1 studies are not really efficacy studies at all. A phase 1 study most commonly involves men who are generally in the range of 18 to 25 years old, who are very healthy. So why men? Why that age? Because they're going to be receiving a drug. We don't want to give the drug at this stage to anyone who could even potentially become pregnant or who has other health-related issues going on. And the only purpose of a phase 1 study, generally speaking, is to get some early safety data and to study what happens when we administer the drug in the body, meaning how quickly does it get to the bloodstream? How quickly does it get to the target tissue? These are called pharmacokinetic and pharmacodynamic studies. They really don't have anything to do with the effectiveness of the drug to treat a given condition. It might help with what is the dose that we're going to use in a future clinical trial to treat that condition, but initially this is just healthy men ages 18 to 25. We then have phase 2 studies, and I'll link that to phase 3 because they're very similar. A phase 2 study is a test study. It's a smaller scale. Phase 3 study, there is an element of efficacy now, so we are treating the target population. Maybe that's people who have relapsing, remitting multiple sclerosis, for example. We're still going to get safety data. We always collect safety data. Usually they're what are called primary outcome measures, the main thing that needs to be studied, and then there are secondary measures that are added on. The phase 3 study then is the larger version of the phase 2 study. That's the big study that's going to be used if the treatment is deemed successful or effective, and then the company that owns that product is going to submit their data to the FDA for approval. There are also phase 4 studies, and these involve what are called post-marketing studies. So we're gathering additional data about the drug or treatment, but the drug is already approved and out there in the marketplace. Now, I've seen stem cell trials where subjects are told that this is a research trial, but they're actually being asked to pay for their treatment. And the FDA generally frowns upon subjects being asked to pay for their own participation in a clinical trial. That's not to say that it can't ever happen. There are some specific guidelines that do allow for this. However, in general, if you are being asked to pay for participation in a research trial, you should be very suspicious. You need to find out more information about why you're being asked to pay. In general, most large clinical trials are funded by the study sponsor. That would be the company that owns the patented product. So in the world of stem cell medicine, that could be Brainstorm Cell Therapeutics that has the Neurone product. It's a mesenchymal stem cell product that is being studied to treat conditions like ALS, Parkinson's, Alzheimer's disease, and multiple sclerosis. So when you go to a website where they talk about, hey, we've got this product and this is our pipeline, a lot of times you can look up and see where is that product in the pipeline. And take the information I just shared with you about IRB approval and phase of clinical trial and see, because if something is in phase 1 trial, remember, that's not even being studied at that stage to treat the condition that might interest you. That's just a safety trial and what we call a pharmacokinetic or pharmacodynamic trial. So how are stem cells regulated by the Food and Drug Administration? In the United States, cellular therapies are regulated by the FDA's Office of Cellular Tissue and Gene Therapies within the FDA Center for Biologics Evaluation and Research. And the FDA distinguishes what they refer to as 351 products versus 361 products. Now, currently, the only stem cell treatments approved by the Food and Drug Administration are 351 products. And they're regulated as drugs or biological therapies, and that would be things like vaccines or neurone and monoclonal antibodies used to treat various conditions. And they have to go through the clinical trial approval pathway that I just explained to you. We have 351 products that are approved to treat things like certain cancers and disorders of blood and the immune system. So the FDA has not approved any exosome, if you've heard of that term, any exosome product for any use whatsoever. So if you have a practitioner in the United States offering to give you exosomes, please understand that is a direct violation of the FDA regulations. Now, by contrast, the term HCTPs refers to human cells, tissue, and tissue-based products. The FDA regulates HCTPs as 361 products under 21 CFR 1271.10a. Whew, that's a lot. While 361 products must meet specific criteria by the FDA to be designated as HCTPs, they are not required to go through the clinical trial approval pathway. These criteria are what distinguishes the HCTPs from drugs and biological therapies. So let's talk about mesenchymal stem cells because they fall under that 361 category. Now, mesenchymal stem cells can be isolated from various tissues such as umbilical cord, endometrium, including polyps, menstrual blood, bone marrow, and fat tissue. And mesenchymal stem cells are multipotent, meaning they can differentiate into a variety of different cell types, including bone cells, cartilage cells, and muscle cells, and fat cells that give rise to marrow adipose tissue. But the differentiation potential of mesenchymal stem cells is not actually what makes them attractive from a clinical perspective. So a little history. Dr. Arnold Kaplan is the scientist Dr. Arnold Kaplan is the scientist who is credited with discovering MSCs. I'll call them MSCs moving forward. So he is known as the father of mesenchymal stem cells. He is the professor of biology at Case Western Reserve University, and he is director of the Skeletal Research Center at the university. He actually proposes that they may be best termed medicinal signaling cells as opposed to mesenchymal stem cells. But if we use the two terms, they are interchangeable. MSCs provide growth factors that facilitate nerve cell growth, repair, and survival through compounds like VEGF, nerve growth factor, and brain-derived neurotrophic factor. MSCs address disordered T and B cell regulatory function. These are your white blood cells. And remember that they not only help you defend yourself against, say, viruses, bacteria, parasites, things like that, but that attack has to be regulated. If it's overly aggressive, for example, then you can end up with autoimmune diseases. So MSCs address disordered T and B cell regulatory function. MSCs increase microglial M2 phenotype by suppressing tumor necrosis factor and interleukin-1 beta. Your brain has its own immune cells, and sometimes those immune cells are protective, and at other times they're on attack. We need them to be on attack at the right times, but we don't want them to be on attack all of the time. And these cells can switch back and forth between their two states. MSCs increase the protective anti-inflammatory type of or form of microglia. MSCs increase anti-inflammatory chemical signals called cytokines. They deliver microRNA that regulate a wide variety of processes involved in immune modulation and neuroprotection. And they are anti-apoptotic, meaning that they can stop or slow the process of programmed cell death. So we see that, for example, in Parkinson's disease, where people affected by this disorder are losing their dopamine-producing cells in the substantia nigra pars compacta of the midbrain. Let's talk about one specific MSC product that's under fairly aggressive investigation, and you can look up all the published papers and go to the website. This is a very legitimate product, the Neuron product, MSC NTF as it's otherwise called, NTF meaning neurotrophic factor. This is from a publicly exchanged company called Brainstorm Cell Therapeutics, originally out of Israel, but they are traded and doing their investigational work in the United States. So how does this process, if you will, with Neuron work? A lot of folks out there with ALS, Lou Gehrig's disease, very interested in Neuron. So in this particular case, if you were a subject in a Neuron trial, you would have your bone marrow harvested. That's because this is one of the places where these MSCs actually live. So under anesthesia, numbing medicine, local anesthesia, maybe light sedation, your MSCs are isolated, are harvested through aspiration of bone marrow, and then the MSCs are separated out from the other cells in the bone marrow. They are then sent off to a central processing facility where they're expanded and then frozen. And then they're shipped back to the treatment location, the clinic, wherever. They're thawed and they are induced to differentiate. And then, Tom, I'm going to repeat this section. Let's talk a little bit about Neuron, which is the mesenchymal stem cell from Brainstorm Cell Therapeutics. This is a publicly traded company in the United States. They have several studies that are ongoing in different stages, the most advanced being their ALS study, and then after that, their progressive multiple sclerosis study. If you were a participant in this study, this is how it would work. Bone marrow is harvested because that's where these MSCs live. So they might use bone marrow from the pelvic bone. They would numb you up, might give you some light sedation, but they use a device that allows them to aspirate bone marrow, and then the bone marrow is expanded and cryopreserved. Eventually, it's sent to a central processing facility where the cells are then thawed and induced to differentiate, and then transported back to the treatment location where the cells are administered directly into the spinal fluid. So we have results from the couple of the clinical trials with Neuron. First, I want to talk about the phase 2 clinical trial, the MSC-NTF, in the use of MSC-NTF or Neuron in progressive multiple sclerosis. This was presented at the 37th Congress of the European Committee for Treatment in Multiple Sclerosis, otherwise known as ECTRIMS 2021. So again, this involved the intrathecal administration of MSC-NTF cells in patients with primary or secondary progressive multiple sclerosis. There were a number of primary endpoints or there were a number, the primary endpoint, I should say, was safety and tolerability, although there were a number of secondary efficacy points, such as how quickly you can perform a timed 25-foot walk, a 9-hole peg test, and other secondary outcome measures. So the bone marrow was aspirated from 20 patients at various local sites, with the aspirate sent to the central facility for culture and differentiation. After shipping that product back to the local sites, about 100 to 125 million MSCs were injected into the spinal fluid in the study subjects at week 0, 8, and 16. There were 46 matched controls. Now, 2 patient participants withdrew from the study because their bone marrow aspirate failed to grow adequately. But of the 18 participants treated, 16 received all 3 treatments, and completed the study, including follow-up at week 28. 2 participants withdrew, one due to procedure-related inflammation of the covering over the spinal cord and brain called arachnoiditis. The primary endpoint, again, was safety. Headache and back pain from the lumbar puncture were the most common side effects reported. There were no deaths and no treatment-related adverse events due to worsening of multiple sclerosis. Improvements were identified in favor of the treatment group for all outcome measures: the timed 25-foot walk, the 9-hole peg test, the MS walking scale, the low contrast letter acuity test, and the symbol digits modalities test. And there was an increase in biomarkers, things that could be measured in the spinal fluid, biomarkers of neuroprotection, and reduction in biomarkers of neuroinflammation. How about the ALS study? This is the most advanced in terms of phases of all the studies. In fact, the phase 3 trial was completed, and Neuron or Brainstorm Cell Therapeutics went on to do an extension trial for subjects with ALS, meaning not a phase 4, but they were allowed to continue to treat the subjects who had already been treated in the phase 3 trial. So this was a randomized, double-blind, placebo-controlled trial. It was announced in November 2020. And I'm going to cut to the chase here on this study because it was very striking. Going back to the phase 2 trial, the treatment group, those who actually received Neuron, had about a 35% slowing in the expected rate of decline of this disease as measured by something called the ALS Functional Rating Scale Score Revised. Now, there was a placebo group in the phase 2 trial, and they had slowing as well. So there's a placebo effect, and this is very common. But their slowing was only about 15% compared to 35.5% in the treatment group with ALS. And this difference between 15 and 35 was considered to be both statistically and clinically significant. And this was the basis for going on to the phase 3 trial. When subjects were enrolled in the phase 3 trial, it's important to know that the investigators enrolled subjects who were more severely affected at the time of treatment. Their ALS was worse. Now, that's very important because when the phase 3 trial was completed, the investigators and the statisticians still found that the expected rate of decline in subjects treated with Neuron who actually had ALS was still about a slowing of about 35.5%. So this matched nearly exactly the phase 2 trial. And there were biomarkers that were being used to make sure that in terms of the spinal fluid, in terms of the cellular activity in the brain, that the treatment was doing what it was expected to do, which was stimulate nerve growth factors, reduce inflammation, slow programmed cell death, and so forth. However, in the phase 3 trial, there was a very big jump in the response of the placebo group up to about 29%. And because the difference between 29 and 35%, which is much smaller, it was not possible on a statistical basis to say that the treatment group benefited more than the placebo group. Now, that's very disappointing, of course, but there's a however. And the however is when the researchers went back and reanalyzed the data and matched those who were treated to be more like the phase 2 trial. In other words, if they only looked at people who were in an earlier stage or a more mildly affected ALS and compared that to placebo, they found the exact same 35 and 15% difference, meaning it's very clear that NurOwn is actually quite effective, but like many other conditions, the most effective intervention is when the problem is caught and treated early. So with that information in hand, not only was Brainstorm Cell Therapeutics allowed to continue with an extension trial, but ultimately they did decide to submit for what's called a biologics license based on that smaller subset of the phase 3 trial, hoping that the FDA would approve the treatment in more mildly affected ALS patients. Disappointingly, the FDA very recently declined that biologics license. However, at this stage, or at the moment, in the very moment that this video is being recorded, that is being appealed. Now, there's more evidence that NurOwn actually works because in South Korea, there is a stem cell product that is very similar to NurOwn that has been FDA approved by the South Korean equivalent of the US FDA. Unfortunately, that treatment is not available outside of South Korea. But in the phase 2 trial, and yes, it was a phase 2 trial, not a phase 3 trial, that led to its approval in South Korea. And we are starting to see a little bit of that in the United States, where conditions that have a great need, like ALS, are getting treatments approved even though they haven't completed all the phases of the clinical trial. We recently saw that with the Amylyx Pharmaceuticals product Relyvrio for ALS. At any rate, the rate of decline, the slowing in the rate of decline of the ALS functional rating scale score of Mesenchymal, which is the name of the product in South Korea, is very similar to NurOwn. Now, we've touched on multiple sclerosis, we've touched on ALS. However, there are other conditions you may be interested in. There is a pipeline for Alzheimer's disease in earlier phases, and there are pipelines for Parkinson's disease as well. So ALS, multiple sclerosis, Parkinson's disease, and Alzheimer's disease, all in various stages of investigation for the use of autologous mesenchymal stem cells in the treatment of these diseases. So let's wrap up this talk so you have an understanding of why it is that under those 1271 guidelines, we don't actually need a clinical trial to treat an individual patient. There's a handful of rules, though, that must be followed under FDA guidelines. That stem cell product must be autologous. It must come from you. And it must be intended for homologous use only, meaning that the cells that are extracted, say, from your bone marrow can't be used for a situation that they would not normally be used in. So we know that in situations where there is injury and inflammation in the brain, MSCs migrate from the bone marrow, generally from bone marrow found in the skull, and go into the cerebrospinal fluid. And there they are activated and express neural elements, and they aid in the repair of tissue. They affect the behavior of those microglia and so forth. So that's homologous. In the United States, any stem cell product that is being used, even if it's autologous and homologous, must be minimally manipulated. So we can't, outside of a clinical trial, so very important, if it's in a clinical trial, that's different. Outside of a clinical trial, we cannot manipulate the cells. We can't do what Brainstorm Cell Therapeutics does with NurOwn. That is under a clinical trial. So if I extract your bone marrow, separate out your MSCs, intend to give them back to you, I can't manipulate them. I can separate them from the rest of the cells. That's called minimal manipulation, but I can't do things to your MSCs before I give them back to you. We can't combine the MSCs with another article. In other words, there's been a temptation to, say, use drugs that cause the MSCs to multiply. I can't give you that drug. I can't mix the stem cells with another drug. I have to use pure stem cells. We cannot use a stem cell product that would have a systemic effect. So there are companies out there that will extract your bone marrow, separate the stem cells, and then dump them into an IV bag, put an IV catheter in your arm, and give you IV stem cells, intravenous stem cells. That is a direct violation of the FDA guidelines, because that would be considered systemic treatment. And finally, the stem cell treatment cannot be dependent on the metabolic activity of living cells for its primary function. In other words, the stem cells themselves have to do the job. They can't in some way be inert and then another part of your body or metabolic activity then somehow makes them active. Now that's a little bit of a gray area, it turns out, because cerebrospinal fluid itself acts as a proliferative niche for neural progenitor cells. What does that really mean? And this is really the crux of the talk today, and I appreciate you listening to this and following along with me. We can't give you NurOwn unless you are a participant in a NurOwn clinical trial, and we are a study site for Brainstorm Cell Therapeutics. Remember that NurOwn is a manipulated MSC product. However, NurOwn, there's sort of a black box, because this is what makes NurOwn proprietary. Those cells get shipped off to a central processing facility, and we don't really know what is done with them, but we know that something is done with them to cause them to express these neural elements that then in turn make these stem cells know that, say, they're in the brain, they're in the spinal fluid, they're going to act on injured brain tissue, let's say, as opposed to injured muscle tissue. It turns out that your own cerebrospinal fluid is more than just a cushion for the brain, more than just a cushion for your spinal cord. Cerebrospinal fluid is metabolically active. And we're not manipulating those cells ahead of time, but if I put those cells into your spinal fluid, your own spinal fluid will cause those cells to express neural elements and will in turn cause them to behave as if they are native to the central nervous system and act on nerve cells and other cells, the microglia. And so it's the environment, the stem cell, cerebrospinal fluid interaction that really may pave the way for cell-based therapy in neurological diseases. So I hope this has been very helpful. I would invite you to reach out to me, to reach out to my clinic. We have a wonderful website, charlinregeneration.com, that has a contact form in it. I want to tell you a little bit about what happens if you do come and see us for stem cells. We are first and foremost going to emphasize that this is an investigational treatment. I've just explained this all of that to you in this video. Stem cells, again, are not proven yet to treat any of these diseases, but we do know that they have a specific role to play in the body, and that is what we talk about. Repair of injured tissue, anti-inflammatory, anti-apoptotic, secrete nerve growth factors, and microRNA. So if you come, let's say you've decided that you understand this is investigational, but you have ALS, you have multiple sclerosis, you have Parkinson's disease, and you would like us to treat you. Once you're checked in, we're going to make sure that you are well hydrated. The day of the procedure, we're going to lightly sedate you. We're going to use local anesthesia, and we have technology from a company that has FDA clearance for the use of their technology to aspirate bone marrow and separate stem cells using what is called a closed system. And what that means basically is many stem cell products require a biologist or technologist and what's called a hood, because they are using test tubes and pipettes and so forth and microscopes and separating out cells. When you have that kind of setup, it does introduce the possibility of infection. We're keenly aware of that at Sharlin Regeneration Center, and by using a closed system, it means that your bone marrow and your stem cells never touch the air, never touch human hands. They are always within a closed, sterile tube. Now these tubes, once we've collected the bone marrow and we use a centrifugation process to separate out the MSCs, we then bring you to a different room, another treatment room where you're turned on your side. What's called the L4-5 interspace is identified. That's the lower lumbar area away from the spinal cord. It's called the lumbar cistern, where the spinal fluid collects inside the spine. That is accessed after numbing you up, once again, having you on your side. This is a standard lumbar puncture procedure. We perform hundreds of them every single year for diagnostic as well as, in this case, therapeutic purposes. And those stem cells are then introduced directly into your spinal fluid, and that takes just a couple of minutes. And then afterwards, you're put into a position that encourages those cells to migrate toward the brain. It's a very easy procedure. Overall, it's relatively easy on the patients. They go home after a couple hours of rest and monitoring in the clinic and come back the next day to the clinic so we can double check everything, make sure you're okay, take a look at the incision site where the bone marrow was aspirated, take a look at the lumbar puncture site, and so forth. If you're okay, generally folks are going home the day after that. We do also prescribe medication for your comfort throughout the entire experience. We make sure that you have direct communication with myself and my staff throughout that time. We're here to monitor you and make sure that you are safe. We've done lots and lots of these procedures. I'll be making a very similar video with some testimonial, and as well as so you can actually see a patient who was treated and see that process. But I hope that this was very helpful to you. All of this, again, will be presented with PowerPoint visuals for you. But if you're very interested in what I've just shown you, please check out sharlinregeneration.com, SHARLIN, regeneration, REGENERATION.com, or give us a call at 417-485-4330. I want to thank you for your time and attention.